Okay, let's continue with fungi part 2. Here we will talk about the form, nutrition, and reproduction that make fungi distinct from other organisms. When you look at most fungi, this is what you and I often see. We're looking at a reproductive structure, or a mushroom. These mushrooms are often quite poisonous, and one must be very careful in picking mushrooms, and even more careful when deciding which mushrooms to eat. Let's look at the morphology of that mushroom body. The mycelium is the body of the fungus. That's an underground matrix of hyphae that are widely dispersed. With some signal, these hyphae come together to form a stalk, which then results in the production of a fruiting body. The fruiting body, the stalk, and the mycelium are all composed of hyphae that have banded together. And recall that a hyphae is a long cylindrical structure closed at both ends with many nuclei within, and those nuclei are separated by septa. We can look closer into the septa in this highly magnified image on the left and see the septum, or a plural septa, that separate the different nuclei. And on the right we see that in this septa there is a small pore, and through that pore or opening, cytoplasmic fluids can flow. Fungi have both asexual and sexual life cycles. Most of the fungal life cycle is spent in the haploid or 1N stage. Below, we have an image of the sexual life cycle, which involves two mycelia that come together and form a diploid zygote. That zygote is the only stage in which the diploid form exists. Let's explore this a little bit further in the next image. Here on the top panel, what you see is hyphae of different mating types coming together to fuse. That fusion is called plasmogamy, and a small chamber is formed in which there are multiple nuclei from each of the different hyphae. A wall is formed called a zygosporangium, and inside the zygosporangium, the nuclei fuse to form diploid zygotes. Once formed, meiosis takes place almost immediately with the production of a new combination of haploid nuclei, which then are dispersed as spores. So spores play an important component of both the sexual life cycle and the asexual life cycle. It's important to recall from before that a spore is an inactive form of life. It is a resting stage capable of surviving for many years in this dry form. If we look at this in cross-section through the soil and through the mushroom, what you see is that below ground, fungal hyphae come together for the process of reproduction, or sexual reproduction, as shown in the previous slide. When some signal, which I don't know, comes about, these mycelium band together to form the reproductive structure. Coming out of that reproductive structure are spores. So we have haploid forms that are produced as spores, which can also serve as a resting stage. Notice that in this conversation of mycelium and hyphae, in some sentences hyphae ends with an E, and otherwise it ends in an A. Hyphae with an E is the plural of hypha. Remember that both the reproductive structure and above ground and the mycelium, the below ground widely spaced hyphae, are dense concentrations of hyphae, shown here on the right. So all of these are the same kind of structure. They are multinucleated organisms called hyphae. As it turns out, not all fungi produce hyphae. One of the derived groups that does not produce hyphae are the single-celled fungi known as yeast. They are smaller in size than our hyphae, appears as balls, and they asexually produce by budding. On the left image, you can see some budding beginning to occur. So keep in mind that fungi can exist as single-celled organisms with a single nucleus, or as multicellular fungi with web-like bodies called hyphae that have linked together to form mycelia. Bryn Dentinger on the University of Utah faculty here in biology studies mycelia of Amarillo solidipes in Oregon. He discovered that a single mycelium can be very, very large. In fact, he's documented that one of these mycelia in Oregon spans more than 2,000 acres. In Bryn's terminology, that's one humongous fungus. Let's once again review this asexual life cycle, which is the most common in fungi. Spores fall onto the ground. If there is water and nutrient available, that triggers germination of the spore, which then produces a hyphae. Those hyphae come together at some point to form a mushroom, which is composed of multiple hyphae banded together. At this point, let's introduce the observation that fungi have different feeding patterns than we find in animals. In fungi, the cell wall prevents the process of phagocytosis from allowing hyphae to take up external food. So what happens is that hyphae now excrete enzymes that break down the organic matter adjacent to the hyphae. Then what we find is that the hyphae grow to this decomposed organic matter and take up these small molecules, and in a sense they are growing whenever they are eating. We can grow hyphae on decaying wood, or we can bring them into the lab and feed them and watch them grow on an agar plate. Remember that hyphae are heterotrophic, that is that they depend on external reduced carbon sources, And they produce CO2 through mitochondrial respiration, just like animal systems do. That means that hyphae need oxygen in a manner similar to that we found in other eukaryotes. Since hyphae have cell walls, let me just repeat that they cannot engulf food in the same way as organisms that do not have that. So they have this absorptive nutrition, in which enzymes are secreted to digest large food particles. The enzymes are produced in Golgi, the Golgi apparatus, which then forms small vesicles carrying the digestive enzymes that make their way to the surface cell wall of the hyphae. They are excreted into the external environment where they break down organic molecules, and those small organic molecules are taken up, and as they are taken up, growth occurs. So let's once again review this very different form of nutrition called absorptive nutrition, in which enzymes are secreted outside the cell to digest food. First, in the upper image, number 1, we find that vesicles carrying digestive enzymes fuse with the plasma membrane, excreting then the digestive enzymes into the external environment, which then breaks down organic polymers. The small molecules that are then formed are taken up by the hyphae, and that results in growth. The higher solute concentration that occurs following the uptake of these molecules causes the hyphae to take up water via osmosis. And as a result of taking up the water, the hyphae can grow. At this point, let's stop the video and test your understanding of what you just learned with these 3 true-false questions. After you have written down your answers, restart the video to learn the answers. Here are the answers to the true-false questions. False. Mushrooms are not diploid fruiting bodies in fungi. Mushrooms represent instead a haploid part of the life cycle, similar to hyphae. Yeast are a single-celled fungi. True. Most fungi are heterotrophic and use phagocytosis to absorb food from the environment. This is false. Fungi have an external digestion system. Phagocytosis is associated with an internal digestion system. This ends part 2.